Sir, should I present? Should I share now?
Sir, yes, sir.
Yes, sir. Yeah. Okay. Okay. Friends, is it okay? Is it okay, sir?
Yes, sir. Yes, sir.
Okay. Okay. So this is title of my today's presentation, and I will talk about near about 45 to 50 minutes. Then the topic will be open for discussion. So, friends, everybody is familiar with this bird. It's vulture, and I have seen it last 25 years ago, and now they are on a edge of extinction. What will be the reason? Whether is it a pesticide or diclofenac? Because why I am saying this? According to one study carried out by one Pakistani scientist, probable reason of decline of this bird is diclofenac. It's a drug. It's analgesic, antipyretic drug, which is used not only for curing the humans, but it is also used for vigor in case of animal, and probably due to eating of meat of that dead animal, this bird is on the edge of extinction. Friends, no doubt, we are now self-sufficient due to green revolution in India. But its darker side is remain obscure. In the process, we have poisoned our soil, water, and even air with pesticide. And we are all familiar that high yielding crops can be sustained with the help of fertilizer, new farming technique, and especially extensive use of pesticide. Friends, pesticides are used to control pest ranging from weeds to different kind of animals, starting from insect to mammals. And according to one study by Nayak and Solanki in 2021, currently about 104 pesticides are being manufactured in India. Along with there are about 293 registered pesticides. And these pesticides are they are widely used. We are all familiar that pests are the organism that compete with man for food, shelter. Now, most of the pesticide used go waste, and they are used indiscriminately. Friends, the chemicals which are used to control pest are includes pesticides, and pesticide includes a number of other chemicals: insecticide, weedicide, fungicide, rodenticide, etc. In habitat, we used to control the target organism, but all other target non-target organisms they are suffered, because pesticide cannot distinguish between the organism we want to control and those we don't want to control, and can kill all of them. According to one study, it is estimated that 1% of pesticide applied reach the target organism, and remaining 99% get incorporated into the ecosystem, which is get turned poison. Their estimation was based on study where caterpillar of one Eris repi had infected the crop at about 1,500 per hectare. Pesticide residues are reported world over in penguins in Antarctica and in oceans everywhere they are reported, the residue. And we are familiar that the ultimate sink of pesticide is water. They run up, they reach by runoff or by subsoil movement of water and may travel thousands of kilometers away from the place of application. Persistence is yet another factor that is concerned. They accumulate and biomagnify in the bodies of organisms. Fish eating birds have a highest concentration than grain eating birds. It was estimated. When we talk about bird, many bird program their life histories to match the time of insect availability. Since they are high in food chain, they may reflect pollutant hazardous to human better than any organism. Females can excrete metal into it, but most of the are excreted from molting of feather. There are a variety of non-lethal effect on birds. For example, their eggshell becomes thin. There is a cholinesterase inhibition. There is abnormal reproductive behavior, delayed ovulation, reduced cluster size, abnormal construction of nest, aberrant incubation behavior, less heat applied to egg being incubated, reduced hatching success, abnormal behavior in chicks, abnormal parental asperia, aspermia in male, susceptibility to predator increases, abnormal development of cheeks.
टननन कहाँ बजा है? हम तो टननन एको बार नहीं सुने टननन बज रहा है।
Abnormal beak in chickadee, one can see in figure, and one pelican. This may be due to contamination. Decline in population create ecological imbalance, and we are familiar about sparrow, example of sparrow in China. So far in birds, there is no immunity against pesticide, but insect pest they have developed. Effect of exposure of variety of pesticide are not known. Thailand has lost most of its bird due to excessive use of pesticide. And this is the fact that the field was sprayed by pesticide and birds are eating the insect and they are used in heavy amount. Even now in India, they are not used in this large scale, but quite long ago, such a type of pesticide appliances were used to control the pyrilla infection in sugarcane, and they are used in a vast variety. Ultimately, what happened? The uh, secondary poisoning can result from mass treatment. If we treat the field, then the insect they get killed, and such killed insect, when taken by bird, ultimately the effect has seen. Birds are unaware about the danger of pesticide because they are eating their food material, but they are not familiar that the food insects are killed by dangerous pesticide. A pesticide ultimately end in nearest water body, and therefore highest amount of concentration of pesticide is reported in aquatic birds, especially. Bird consume vegetation sprayed with pesticide. Due to that, they may get affected. Pesticide killed or impaired insect transmit pesticide from that ecosystem into their body. Insect killed by pesticide are fed by bird to nesting, and due to this, this is the one photograph of sparrow which is feeding their young one by insect. Carnivorous bird consume the herbivorous bird, and that's why the pesticide again get into the carnivorous bird. And the most important effect which is seen is biomagnification, because concentration, this is one figure which can explain. For example, concentration of DDT in water, it is about 0.03 ppm, and when it goes via plankton, via food chain into the carnivorous bird, it may increases up to 25 ppm. So this is biomagnification and bioconcentration is the effect of the food. Uh, whether Sarus crane, a beautiful bird, it may follow a path of vulture. And we are familiar that central government has taken an initiative to conserve the vultures by starting the vulture restaurant, which is near in Nasik. In sanctuaries, the main source of pollution is a vehicle, because the animal, they are when cross the road, they e- automobile exhaust, it directly goes into them. Herbivores frequently the same road as do vehicle that emit exhaust. An animal that venue out of this road has to inhale exhaust left by the vehicle. Many mammalian species like chital, nilgai, sambar, wild boar, hare, porcupines are known to enter agricultural crop and fed on vegetation which has been sprayed with variety of pesticide. Carnivores that fed on these are animal get pesticide due to biomagnification. Sometimes these wild animal are intensively poisoned. Particularly vulnerable are the big cat, as they kill cattle and in retention villagers may poison the carcasses of the kill. Poachers are also prefer the method of poisoning as they get undamaged skin of wild cat. This is the figure of poachers. They are carrying the uh, animal. Treatment of forest with calcium arsenate from aircraft started in 1920, and rabbit kill were reported during that time. Wild mink from agricultural field in Iowa had residue of DDT, DTE, TDD in their adipose tissue. Similarly, deer from Arizona who had a residue of TDT and its metabolite and that of aldrin-2. Unfortunately, there is little work on mammalian toxicology in India. Metal and bird. Bird can excellent qualitative indicator as well as quantitative monitor of environmental pollution, particularly air pollutions. Since they are mobile pollutant, are since they are mobile, pollutant are picked up from wide, often ill-defined area. Many of them share the habitat with human as commensal. Bird population changes in response to altered habitat. Population also decline if enough insect are not available for feeding to chicks. Level of metal in soft tissue, blood, bone, feather, egg, and fecal matter are used in monitoring. This diagram will show you, this picture will tell you what is the effect of heavy metal residue, and it reduces calcium source, decreases number of caterpillar, ectoparasite. As a result, there is a thin cell in egg, small cluster size, poor hatching, and nesting mortality is reported in bird. Metal level in faces of bird indicate daily exposure of bird to metal. This can be used to monitor pollution level in bird colonies, in sanctuaries, or in zoos which are located in urban settings. Several studies indicated exposure of wild mammals which are living near highly contaminated area like smelter, uh, chloralkali plants, heavily used highways and mines. Many wildlife species like raccoons, cheetahs, sambar, wild boar, and nilgai ingest soil while feeding and may accumulate metal present in the soil. Most vegetation is known to accumulate metal. Significant concentration of lead was reported in mule deer and prognor antelope feeding on roadside vegetation. Friend, ancient skeleton as a silent witness of lead exposure were studied in Scandinavia. Carnivores, teeth, and antlers have also been studied for metal accumulation. Mammalian fecal were used to study exposure of wild mammal in zoos and various sanctuaries of Rajasthan by Gautam. So one cannot avoid the use of pesticide, but at least one can go for integrated pest management. And due to use of integrated pest management, both the pest population will be reduced and there will be no effect on animal and other ultimately on mammals. So friends, I am going to finish my presentation and the topic is open for discussion. I express my sincere thanks to all. Uh, yes, sir. Hello.
If any participant wants to ask any question, it is welcome.
The topic is open for the discussion.
Hello, sir. I'm audible.
Yeah, madam. Please.
Uh, Respected sir, I have one query. What is
Yes.
the integrated pest management to control pesticide? What is the role?
Yeah, madam. Integrated pest management involves use of mechanical control,
Okay.
prevention, biological pesticide by use of these all other methods. So
Okay.
one can reduce the use of pesticide.
Okay. Okay. Okay, sir. Thank you, sir.
Okay, madam.
Sir, there is
Hello,
in
sir.
Yeah, yes, madam. Yes,
Sir, I have one question to you. So,
yes.
what is the probable effect of this pollution on cellular, molecular, and genomic level of the organisms nowadays?
Yes, madam. No doubt. When pesticide enters in the body of any organism, the mechanism of action is given and it affects directly up to the cell level, up to the nucleus. Um, so there is a separate branch which is known as genomic toxicology. So you can go for that. It's available.
Okay. Okay,
Okay.
sir. Thank you. Thank you very much,
Yeah,
sir.
yeah,
Thank you.
yeah, yeah.
sir, I have a question. May I?
Yes, sir. Yes, sir. Please.
So there is any ratio that normally present in the environment, the pesticide, and then ultimately transfer into that accumulation into the bones or other. There is any ratio. There is such type of amount may present in water that from this particular amount it transfer normally their bones or other tissue. There is any ratio.
I haven't gone for that study, but it is available, sir. Because what happened when pesticide from water it goes into plankton, then aquatic animal, and at higher level there is biomagnification and bioconcentration when it goes in body. Because see, sir, DDT, BSC, they are banned quite long ago. but till today the residues are found. So it may be there. Yeah.
Hello, sir.
Yes, sir.
Sir, I have one question.
Yes, yes, sir.
What
Please.
are the obstacles to minimize the use of pesticides?
What I think, sir, I think so. Awareness is essential because I am from the area of Nasik district, and which is famous for cultivation of tomato, cabbage, and say grapes. Uh, the farmers they are not familiar. Even before harvesting, 2 to 3 days of cabbage, they bring it within the field market. So I think one can reduce the use by awareness, and there is a demand of organic food material. Nowadays, there is little awareness, but one can go for it. And some government policies are to be required for that. I think so. Yes, sir.
So if there's any relation or any kind, any information regarding the pesticide and microbial growth, there is because the some pesticides become if they are degraded, so after degradation they are anoxic condition. So this can introduce the microbial. So there is any such type of work or relation that can relation to the pesticide, which may be normally degraded, not the biomagnification, but degraded, but that ultimately relation to the microbial activity.
Yeah, yeah. Sir, what happened? I think many pesticide which are available, they are not degradable, and therefore many of them they are now banned. For example, even endosulfan, which are used, it is banned. So the area research area is very good for next generation. If any microorganisms were developed which can degrade the pesticide, ultimately it's good for welfare of all mankind. I think so. But till today, there is no available of such a microorganism which can kill the or degrade the especially pesticides, according to my knowledge.
Okay, sir. Thank you, sir.
Okay, okay.
Are there any other questions?
Yes, please.
I think questions are over.
Yeah, yes.
So let's conclude.
Yes, sir.
On behalf of the organizing committee,
Yes,
My
sir.
Education Society's Arts, Commerce and Science College, I show my gratitude and thanks to Dr. Sanjay Nikam, who is also my best friend. Since last 10 years, we are working with Board of Studies in Zoology. Sir, we are thankful that you are enlightened and focused on the environmental pollution, which is really the important issue nowadays, as chemical fertilizers are banned and we are going towards the biological control methods. So, on behalf of More Education Society, Sonai Arts, Commerce and Science College, and the organizing committee, I am thankful to Dr. Sanjay Nikam, who has taken out 1 hour through the tight schedule of these elections in Maharashtra. So, thank you, sir.
Thank you, thank you. So
So,
yeah, I express
yes,
my
sir. Yes,
sincere thank to Dandavate sir and Sheikh sir, due to which I got on this opportunity. Thank you, sir.
yes, sir.
May
So
I
this
leave now?
session has been concluded.
Yeah.
We'll proceed for the next
Yes.
presentation. Thank you, sir.
Okay. Thank you, sir. Thank you, sir.
Thank you, sir, for enlightening us. Now I request all the participants to kindly join their respective group links for the further presentations. Group 2 participants from group 2 can stay on the same link for their presentation, and for group 3, 4 and 1 we have already shared the link in the group. Kindly join the link. Participant from the group number 2, zoology. I hope everybody is from the group 2.
Yes, sir.
I request Dr. Khaide sir to please start the session. Those who are ready with the presentation can start with the presentation. Can we have any volunteer from the group?
May I, Dr. Sanjeev sir?
Yeah. Are you ready, sir?
Yes, sir. I'm just sharing this. The option is not 1 minute. Screen share. Okay. Screen is visible, sir?
Yes, sir.
Okay. Okay. May I start, sir? May I start, sir?
Yes, sir. Please start.
Good morning, Honorable Chairperson and respected all. Myself, Dr. Sanjeev Shah, Assistant Professor in Zoology, Vidyasagar College Forum and Calcutta University. Today, I am presenting a paper entitled Impact of Climatic Change of Prevalence of Waterborne Vibrio Species and the Risks of Zoonotic Transmission in Fisher Person of Indian Sundarban. So it is changeable slide, sir, or not?
Not, sir.
Okay. So all of we know that the climatic normally the average minimum more than 30 years of weather condition normally we called as the climatic, and this climatic system there is a 5 component: atmosphere, hydrosphere, cryosphere, lithosphere, and biosphere. So interaction between this complex component normally that determine the climatic pattern. And this climatic pattern, long-term change or significant change of this climatic pattern are normally known as the climatic change. So if you see this table, there is a five kind of climatic pattern determined by the Köppen. One is the tropical, dry, temperate, continental, and polar. And this is this slide showing that the NASA in NASA scientists shows that or already reported that the more than in last 100 years up to the 1975, the average temperature increase only 1.1 degree centigrade, whereas after 1975, each decade or the after each 10 days, the average temperature increase from 0.15 to 2 centigrade per decade. And this slide showing that, first is showing that there is a highest the temperature anomalies during this 2022. And within these 10 greatest anomalies country in temperature, India normally as a India situation is the 7th position out of 10 hottest country. And if you go through this 2023, it is the warmest year within this. With 23 is the warmest year due to their long climate anomalies, 90% energy absorbed by the ocean, as well as the El Nino effect. Next slide. So this slide showing that the anthropological activity normally increase the greenhouse gases, and within the greenhouse gases, if you see the first picture, this carbon dioxide is the main component that increase the highest temperature. So ultimately increase the carbon dioxide and other uh, greenhouse gases ultimately alter this climatic pattern, and this pattern long-term change ultimately change the climatic. And after the climatic change, these are the effect or the normally there is a rising temperature, rising acidity, and that means pH, alters salinity, rising the level of the sea, natural disaster. So ultimately collection, this kind of total activity increase the multiplication or the reproduction of the vibrio. And after vibrio and increase the vibrio population, then the normally the in case of natural disaster as well as the flood, that can contaminate the natural fresh water as well as the animal. So ultimately through this climatic change ultimately in contaminate as well as increase the vibrio population for their suitable environment. Next slide. So next slide is the showing that the normally the vibrio, the in case of they are non in case of in adverse situation, that is low temperature, salinity, in such case they are not present within the environment. In that case they normally situated within the normally the animal that is normally in the ocean or sea, that is the fish, sea fish, and normally the shellfish, that is the crab, prawn. So normally in adverse situation they situated within this particular species. So when the fishermen normally collected these species from the natural habitat, they are contaminated directly. So normally contamination area, there is a wound and rough handling, or as the when they are cooked, when we are also cooked or the use this without proper preparation, then we also contaminate with the particular vibrio. So this table also showing that the normally after of the 200 infection disease, normally 12 vibrio are normally they are uh, infectious or the zoonotic in nature, and among them Vibrio vulnificus, Vibrio mimicus, and Vibrio vulnificus, cholera, and the Vibrio paranemicus are the most effective both in the animal as well as the human also. And in case of human, they are normally the problem due to the zoonotic transmission is the gastroenteritis, infection is wound, food poisoning, watery diarrhea, abdomen clumping, nausea, vomiting, chills, etc. So next slide is showing that normally the fishermen are normally affected through these different points. First point is the occasional exposure, because normally the fishermen are close to the nature. So those fishermen are normally affected directly through these points. That means the one is the occupational exposure, wound infection, gastrointestinal infection, septicemia, and economic importance, that is the mass mortality of the fish and shellfish that can reduce their occupational strength. So these are the some pictures that are showing that infection, and in right hand side the picture showing that the transmission of zoonotic transmission of the Vibrio from the nature up
ये
to
तो कल तो सृष्टि ने भेजा था
through
वो
the
वाला
up to the
सृष्टि होगी इसमें तो समझ जाएंगे कि सही है सृष्टि माहेश्वरी नहीं होगा तो फिर उसको फोन करेंगे
And
हम
next
लोग।
slide also showing that the temperature in the climate change ultimately increase the contamination of the Vibrio, and this contamination ultimately to the food poisoning, increase the food poisoning, diarrhea in case of human, and especially in the fishermen of West Bengal. So these are this picture showing the different site for collection of the sample. And after collection, we normally detect the Vibrio concentration.
ऐसा हो रहा था कि जो
Physically
सुनने
as
वाला
well
था
as the
वो बस सुन ही
determine
रहा था वो पूछ रहा था
physically
कि
as well.
कोई क्वेश्चन उस पर से 2-3 के पास उसने पूछना भी बंद कर दिया। हाँ तुम बताओ वो बस लोग
Yes, sir.
रीडिंग कर रहे थे इतना अच्छा मौका था।
May I continue? Sir, may I continue?
Hello.
Hello, may I continue?
Yes, please. I request all the participants, please mute yourself.
So we first
Samreen, Manisha Patil.
First we identify the Vibrio as a physical and then the molecular level. So these are the procedure. Firstly, collect the sample as well as the water from this selected site, and then transfer into the culture media, then purify, and after this purification we growth in the TSA, the tryptophan soya agar, and then the broth, and some part of the collection for further use in the storage, and some are directly used for our identification. That is the, if you see this kit, through this kit identification we can identify the different type of Vibrio as a physical manner. So these are normally after the plating with the we count this Vibrio in different part: water, carapace, in shellfish, gill, hepatopancreas, muscle, blood, and around among these the normally the blood the collection is the high in case of the monsoon season, because there are monsoons, there the multiplication is very high and contaminate very well. So it is this particular this result showing that during the monsoon the infection of the Vibrio is very much high. So this is the molecular strategy. So after collection, after giving taking the particular broth, and then we transfer into the pellet, and use the pellet, then you transfer we use the enzyme reaction and to transfer collect the DNA, and then finally the after particular specify within the specific machine, there is the nanodot machine the quantity of the DNA, then we use this PCR, and then gel agarose, and ultimately gel documentation. So these are the some primer that we use for this particular identification. So first is the universal primer, then is the Vibrio genus primer, then there are 6 primer used for specific Vibrio related. One is the Vibrio paramoeticus, Vibrio quality, Vibrio vulnificus, Vibrio mimicus, alginoticus, Harvey. These are the normally present in the West Bengal coastal region. And lower portion is showing the toxic gene or virulent gene that can transfer from the natural habitat to particular fish or shellfish, and then transfer into the human. So we identify normally the 3 kind of toxic gene. One is the TOXR, then TDH, and then the TRH. These are the toxic gene of Vibrio. So these are first slide showing the bacterial universal bacterial pattern, and second slide showing the presence of the exact Vibrio species, not a specific, but the genus Vibrio, presence of genus Vibrio in different collection site. And this slide showing the presence of Vibrio vulnificus, alginoticus, pyotemicus, and mimicus in different region of South West Bengal or the Sundarban area. And this slide showing the showing the presence of toxicar TRH and TDH gene. So after complete this portion, we knowing how to act the function as a zoonotic transmission. So for this purpose, the zoonotic transmission, we use the rat group into the four different part. One group only use the dead Vibrio, that is the heat killed Vibrio. Second is only the live Vibrio. Third one, the live Vibrio with antibiotic directly proportion 1:1. And fourth one, normally we use the 3 doses of antibiotic before entering the Vibrio. So this is the result. In case of dead Vibrio or the heat killed Vibrio is the 99 point something, 99% is alive, and 1% one or less than 1% is dead. In case of Vibrio, it is the 100% within 24 hours. All these are reported in 24 hours. In case of second one, the 100%, the mice is become died. In third, there's a 90% died and 10%, sorry, 90% alive and 10% died in case of use the Vibrio as well as the antibiotic. In fourth case, when he use the 3 doses of antibiotic and then use the Vibrio, then near about 100%. So heat killed and the 3 doses of antibiotic giving the near about same result. So after this experiment, we collect the blood and we plate it in the TCBS culture medium, and these are the pictures showing the, in case of live Vibrio, then is the 100% dead, then the 10% is dead, and this is there is no Vibrio, that is 100% is alive. So this is the ultimately the result. And similar result also giving in case of enzyme. We collect the serum after the we use the serum of the mice, and we ultimately 3 kind of experiment we have done. The one is the GOT, second is GPT, and third one the alkaline phosphatase. So if you see this result, the control value as well as the mice that are ultimately giving the 3 doses of antibiotic and then use the Vibrio near about same. These are 33, these are 53. Whereas with the mice with direct Vibrio, mice is Vibrio, that is the that means the 100% died. In case of serum, it is a 545. It is a more than 100%, near about 100%. Whereas mice use the antibiotic, in that case, particular GOT is the 109. So there is some recovery giving recovery seen in case of when you use the antibiotic with the Vibrio. But in case of if you use the antibiotic before the Vibrio, then it is the near about control value. So same case we have seen in case of SGOT, SGPT, and as well as the alkaline phosphate. In case of alkaline phosphate, 97 is control, whereas 137 is use the antibiotic 3 doses before the use the Vibrio. Whereas the mice without any antibiotic is the 474. So these are the experiment normally used as a there is any kind of zoonotic transmission or not. So these experiment normally we can primarily say that zoonotic transmission has the effect similar as the human also. So prevention, we can prevent through the use that in case of fishermen, they use the different kind of, they can firstly the prevention means they can use different type of prevention gear, protecting gear, ensuring the proper wound care or not. There's one part, the wounding through this, the infection through this wound. So if you properly care with the wound, then we can ultimately prevent this disease. So second is the vaccination. Third is the monitoring and reporting. So from this, we can conclude the within this out of the 12 Vibrio, normally Vulnificus and apart from this quality, Vulnificus and pyrometric is the most enteropathogenic Vibrio that cause zoonotic transmission in shellfish as well as the man. There is the fishermen, mostly fishermen, those are normally located near the nature and directly collect the specimen. So Sundarban is a reservoir of the enteropathogenic bacteria from this toxic gene may cause the devastating condition in the future. The seasonal physicochemical water quality farm may use as a early warning signal, and the proper management strategy can reduce this vibrio infection. So these are some our publication, and we are the thankful the West Bengal government of West Bengal DSTBD funding support the project is a part of our total project. The main project is a diversity and impact of zoonotic strain of Vibrio in aquaculture skylight in West Bengal. This paper, this part is a main project, and I am discussing here in one of the part of our main project. So, thank you, sir. Thank you, sir.
Are you aware about antimicrobial proteins?
Now, say I have also there is another project is submitted there in this particular project. We are also working on the particular protein that can ultimately infection. There's another part. I just starting this for 2 years. This project for 2 years. Within 2 years, I ultimately this brief portion we can discuss in not in further we can discuss in detail in other project.
Okay, proceed further. Nice work.
Thank you, sir.
Next, Jay Shri Shinde. Jay Shri Shinde, ma'am.
Yes, sir. Yes, sir.
Jay Shri Shinde, ma'am.
Yes, sir. Am I audible, sir?
Yes, ma'am.
Okay, sir. Myself.
Shinde ma'am. Hello.
Hello, sir.
हाँ. Please start ma'am.
ओके सर. सर आवाज येत नाही का?
तुमच्याकडून माईक म्यूट होतोय मॅम.
Okay, sir. Okay. My research under the sub-theme agriculture, forestry, and horticulture. Title is an introduction of South American tomato. To absoluta. It is one of the major pests from the agricultural crop kingdom and now, as we know very well, it is believed that insects are so successful because they have a protective shell or exoskeleton. They are small and they can fly. Their small size and ability to fly permits escape from enemies and dispersal to new environments. Nashik is a major growing region. 12,000 hectares of land in Nashik district is used for tomato cultivation every year. The average crop production of tomato in Nashik district is 6 lakh metric tons. The tomato season starts in August to September. Now, Nashik is the major supplier for tomatoes to most parts of the country. The largest wholesale market of tomatoes in Maharashtra is Pimpalgaon one. It supplies tomatoes to other markets like Delhi, Madhya Pradesh, Assam, and Haryana. Also exports tomatoes to other countries like Bangladesh and Pakistan. The ignorance of pest. Total awareness of values absoluta, which is a research experimental pest. Jumbling between other leaf miners and tomato leaf absoluta by farmers. Use of different chemical sprays of other miners and fruit flies for the control of this experimental pest. The methodology we have used a visit to tomato crop fields and survey about the pest awareness in tomato growing farmers through questionnaire. we can show that near about 60% farmers are unaware, totally unaware of the pest. You see the degradation of the crop, destruction of the crop by the pest. To large extent, it loss of tomato crop due to experimental pest infestation and decrease in marketable value. See the normal plant and affected plant. This pest affects a lot. The difference other leaf in first figure we can see the destruction for the infestation
Yeah, yeah.
Hello. Anyone ready with your presentation?
Sir, may I present?
Okay. Kashipa Sheikh, are you ready
Yes.
with your presentation?
Yes, sir.
Okay. Please share your screen and start your presentation.
Yes. Okay. Good morning, everybody. Myself, Kashifa Sheikh, and I'll be presenting on the topic effects of hypoxia on the physiological biochemical blood parameters in fish. Okay, so these are the contents. Okay, so as we all know that the health of the aquatic environment is very important for the survival of aquatic organisms. But with the increasing water pollution, what we are seeing is that the release of heavy metals and fungicides, insecticides, and even natural phenomena like eutrophication, these are acting as environmental stressors, altering the physical and chemical parameters of water bodies. The parameters such as the pH, the temperature, the dissolved oxygen, all of these parameters of water bodies is getting changed. One of the important parameters is dissolved oxygen, and when the concentration of this dissolved oxygen falls below 5 milligrams per liter, it leads to a condition called as hypoxia. And this has a very negative effect on the fish. It affects their growth, their survival, their behavior, as well as their reproduction. What we see in most of the cases is that to overcome this hypoxia, fishes have evolved different mechanisms whereby they will show abnormal behavior, they will show alterations in their blood parameters, they will also show alterations in their biochemistry, such as the carbohydrates, the protein, the cholesterol levels. All of this will alter so that they can overcome hypoxia. So, with the objective of studying what exactly will be the effect of hypoxia on the physiological and biochemical parameters in fish, I have chosen catfish, that is Heterocmustus fossilis, as the animal model. These fishes were divided into two tanks, a normoxic tank, wherein the dissolved oxygen level was 5 mg per liter, and one hypoxic tank where the dissolved oxygen level was 1.8 milligrams per liter. So for a duration of 7 days, these fishes were held in the tanks, and then the blood samples were collected from the caudal vein, and then different hematological parameters were studied, such as WBC and RBC count. The differential WBC count was studied. Total cholesterol, plasma protein, plasma glucose levels, hematocrit, hemoglobin, and the erythrocyte morphology, they were all studied. So the results that were obtained, we saw that in the total WBC count had significantly increased. The RBC, hemoglobin, and hematocrit count had also significantly increased. Now, the RBC count, hemoglobin, and hematocrit count, the increase indicates that the oxygen carrying capacity had been increased. And the WBC count increase in that, it indicates that the immune response of the fish was also stimulated due to hypoxia. We saw changes in the RBC morphology, wherein we saw that the cells had become swollen. Some had fused, as seen in figure 5C. Some had even shown blebbing, and some of the cells had stacked to each other, giving a Rolex appearance. And this was mostly because of the oxidative stress that had due to oxidative stress because of hypoxia. The differential WBC count, we saw that the lymphocytes had greatly increased. The monocytes and neutrophils had decreased, which could be due to the catecholamines, release of catecholamines. Next, what we saw was the plasma glucose level had increased. Now, this could be due to since the hypoxia had caused energy demand had increased. So, to compensate that, the glucose levels had increased, glycogen formation had ceased, gluconeogenesis was taking place. So the plasma glucose level had increased, and because of gluconeogenesis, the cholesterol, total cholesterol levels also had decreased, and plasma protein levels as well as had decreased, so that the glucose level could increase. Okay. So in conclusion, what we saw was when the fishes were put to hypoxia, a lot of alterations were seen in the physiological and biochemical parameters, indicating that indeed hypoxia does affect the fishes. And to overcome that, the fishes had shown alterations in their RBC so that oxygen carrying capacity can be increased, WBC so that their immune system can increase, the erythrocyte morphology. The deformation was mostly due to the oxidative stress, an increase in plasma glucose and protein levels. They reflected that the energy demand was increasing. So with this, so the future aspects of this research can be that since spleen is the organ from where the RBCs are being released, the histology and the spleen somatic index can be further examined. We can also check the catecholamines present for further studies, and we can also see what modifications are happening at the gene level because of hypoxia. These are the acknowledgements. I would like to thank my guide, Dr. Surbi Mishra and Ms. Rutuja Nagod for their guidance, and our HOD, Dr. Satish Saifere. Thank you. The presentation is open for discussion.
What is the scientific name of fish you have selected in your study?
Heteropneustes fossilis.
Okay, thank you. Continue. Best luck.
Thank you.
Hello, sir. Hello.
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 Yes, ma'am.
May I present?
Yes, ma'am.
Oh, thank you. Is my PPT visible?
Yes, ma'am.
Yes.
Yes, ma'am. Yes.
Yes, thank you. Good morning, all. I am Dr. Pallavi Pawar, Assistant Professor at Dr. D.Y. Patil Art, Commerce, Science and Science Women's College, Pimpri. Today, I am presenting my work entitled Species Composition and Distribution of the Aquatic Beetles in Bhima River, Daun Taluka, Maharashtra. Yes. Introduction. The aquatic coleoptera is also called as the beetle, and it is one of the largest order of insects and contains about 40% of known species. The order name Coleoptera is derived from the Greek language. The kelos means the sheath and pteron means the wings. As we know, the insect have four wings and the hand wings. The forewing in Coleoptera, the forewings are hard and the hind wings are membranous and very thin. There are around 13,000 described species of the aquatic beetles in the world, of which 776 species belonging to the 137 genera and under the 17 families and 3 suborders found in India. The aquatic beetles are vital to ecosystem, playing key role in nutrient recycling, serving as a biological indicator, controlling predators, and supporting research, education, and food webs. The present study is focused on the diversity of aquatic beetles of Bhima River from Daun Taluka, Pune, Maharashtra. Material method. The sampling was done from 2022 to 2023 from 6 different localities of the Bhima River of Daun Taluka. The collection of aquatic beetles was done by using the insect nets, kitchen strainer, and hand picking method. The collected species preserve in the 70% alcohol, and the identification was done with the help of literature, and confirm species were deposited in ZSI WRC Pune. The identified species were photographed by using the stereo microscope. These are my localities: the Siddhatek, Alegaon, Daun, Kangaon, Nangaon, Pargaon, and the Ranjangaon, from where I have collected species. Result: total 38 species were recorded from the 6 families of order Aquatic Coleoptera, and these families are Diticidae, Gyarinidae, Hydrinidae, Hydrophilidae, Noteridae, and the Scutidae. Diticidae is the largest family in terms of both species and genera, and it contains 18 species across the 10 genera. Hydrophilidae with 11 species and the 9 genera and family shows a significant diversity next to Diticidae. Gyarinidae is a smaller family with 3 species and the 3 genera. Noteridae even smaller with 2 species and the single and the 2 genera. Scutidae with 3 species and the 2 genera. And the last one is the Hydrinidae. It is the very smallest family with single species and the single genus. The pie chart shows diversity. Diticidae is more diverse family, and the Hydrinidae is the least diverse family, which is found in Bhima River. This table shows distribution of all the species of family Diticidae across sampling localities. It is the largest and the diverse family. Out of the 18 species, the one highlighted in green named Sebistus segillatus, Pistitus, Nilsoni, Hydaticus, Luxonicus, Centricotus digeni are evenly distributed across all the localities, and the highlighted in blue are significantly found only in Ranjangaon area. The next table shows the family Hydrophilidae, Gyarinidae, Noteridae, Scutidae, and the Hydrinidae species distribution. Hydrophilidae is the second largest family, followed by the other families. Here also the green the species highlighted in green shows evenly distribution across all the localities. From the family Gyrinidae,
Respected madam.
from the family Gyrinidae, Gyrinus convexiculus was recorded for the first time. May I continue?
Yes, ma'am, please continue.
From the family Gyrinidae, species Gyrinus convexiculus was recorded for the first time in Maharashtra through this study. This is the table shows diversity indices for the Bhima River based on the beetle abundance data. The taxa indicates that the Bhima River has the 38 distinct species of the aquatic Coleoptera. The Shannon diversity index indicates the evenness and richness of the species, and the value is 3.4, relatively high, suggesting that the high richness of the beetle. The Markgraf richness also, the value 3.7 also shows the high richness, and the equitability shows the high evenness in species distribution. These are the some representative photographs of a beetle of family Diticidea, Gyrinidae, Noteridae, Hydrophilidae, Hydrinidae, and the Scutidae. This is the Gyrinus convexiculus, belongs to the family Gyrinidae, which is the first record of Maharashtra. The study present a comprehensive data of 38 species and 28 genera belonging to the 6 families, namely Diticidea, Hydrophilidae, Gyrinidae, Noteridae, Scutidae, Hydrinidae, under the 2 suborders, Adiphaga and the Polyphaga. Diticidea was found to be the most prevalent family in the Bhima River. The individual of the family Scutidae, Hydrinidae were exclusively found in Bhima River of the Daun Taluka from Maharashtra. Gyrinus convexiculus belongs to family Gyrinidae, recorded for the first time from the Maharashtra. Thank you. Any questions?
Yes, madam. Nice work is there.
Thank you, sir.
What are the favorable factors to increase the density of population of this beetle?
Sir, the nutrient and the temperature.
Okay, thank you. Proceed further. Best luck.
Thank you, sir. Thank you.
Hello. Can anyone ready for present your research?
Sir, may I?
Yes, ma'am. Please.
Sir, may I?
Yes, ma'am.
Oh, just 2 minutes.
Sir, can I start?
Yes, ma'am.
So, good morning, everyone. Myself, Padma Naik. Today, I am going to present on the topic effect of hypoxia on hepatic enzymes in the catfish heteropenistus fossilis.
सर मी केले होते त्याच्यामुळे सर केले ती
So,
मॅडमचं चालवून गेलं
these are the contents today I am going to discuss. So basically, we all know fish is important part of aquatic food chain, and it is also an environmental health indicator. Any change in the environment, it directly affects the fish. So nowadays, due to excess anthropogenic activities like uh, uh, pollution, greenhouse, increasing in the greenhouse, so these and many more, these all activities affect the environment and the water bodies. So as we all know, DO, that is dissolved oxygen of water body, plays important role for maintaining the life of fish. So when the DO level falls below the 5 milligram per liter, it creates the condition that is hypoxia, and it also affects, due to hypoxia, it affects the fish survival, behavior, and growth, reproduction. So keeping in this mind, so the study was conducted to check the effect of hypoxia on hepatic enzyme. Why the Why the liver? Because liver involves in various important activities like energy metabolism and detoxification. So for this study, we have chosen the animal model, that is catfish Heteropenistus fossilis. For this study, we have maintained the two tanks, one tank in which we have named as normoxia, which is our control group, where the DO level of that tank maintained at 5 milligram per liter, whereas other tank is of hypoxia, where in order to give the hypoxic condition, the DO level was maintained at 1.8 milligram per liter. And after that, both the tanks were maintained for 7 days, followed by the fish was decapitated and the liver tissue from the fish was dissected. After that, the fish liver contains important biomarker enzymes. So any change in this biomarker enzymes will affect the liver. So with this, the liver was further analyze the liver hepatosomatic index as well as the various biomarker enzymes were analyzed. Like in this study, the five enzymes were analyzed like AST, ALT, ACP, ALP, LDH. These enzymes were analyzed by using commercially available kits at and at 530 nanometer colorimetrically. Now, the first, when fish subjected to the hypoxic condition, there are some behavioral changes was observed. The behavioral changes like slowing down movement as fish now under the hypoxic condition. So in order to conserve the energy, in order, so fish activity was slowed down, and there are other activities like crowding, bubble formation also seen, followed by first, the changes in the hepatosomatic and hepatic enzymes. This hepatosomatic index of fish significantly decreased in hypoxia as compared to the normoxia, followed by the hepatic enzyme, that is ALT, significantly decreased in the significantly increased in the hypoxic condition compared to the normoxia, followed by in the other biomarker enzymes like AST, which is aspartate transaminase, and acid phosphatase, ALP, and LDH. They show the significant increase in the hypoxic condition as compared to the normoxia condition. So under hypoxic condition, it was seen that the liver enzymes significantly increased. Now the overall conclusion is that when the fish is kept in the hypoxic condition due to less oxygen, there is a generation of ROS, that is reactive oxygen species. Due to this, what happens? The hepatocytes, the membrane of hepatocytes undergo lipid peroxidation, due to which there is there is there is damage to the lipid membrane, due to which there is increase in the elevated level of AST, ALT, and LDH. This also indicates that liver, due to hypoxia, switch to the anaerobic uh, anaerobic metabolism from the aerobic in order to boost the energy metabolism. And due to hypoxia, what happens? There will be chance, there is chances ki immunity weakened. So in order to improve the immunity, the level of ACP, that is acid phosphatase, and alkaline phosphatase enzyme get increased. So HSI decrease indicates that there is a shift in the metabolism from aerobic to anaerobic. So all these changes brings in order to boost the energy metabolism to improve the immune system of the fish. So the future aspect of this study is that there will be chance further analysis of superoxide dismutase and catalase, and other histological studies of liver will tell how the damage, what are the extent of damage to the liver of fish due to hypoxia, and the and the study of vitellogen protein, and the further if the study of endocrine system, how it affects the growth of fish. So these are the future aspects of this study. So I am thankful to my guide and supervisor, Dr. Surbi Mishra and Miss Rutuja Nigot, and our HOD, Dr. Satish Sarfare. Thank you. Now presentation is open for discussion.
Okay, these enzymes are in the enzymes listed by you.
Yes, sir.
Enzymes listed by you are mostly concerned with cancer.
Okay.
Is there any relation between hypoxia and the cancer?
Mhm.
Hello?
Yes, sir.
Am I audible?
Yes, sir. Yes.
All these enzymes you have been listed, for example, SOD, for example, this
Yes.
enzyme is concerned with cancerous activity.
Uh,
Is there
so
any relationship between hypoxia and the cancer?
till now, I have not found anything about this enzyme with respect to the cancer, sir. So I have to further need on study on that.
Okay, continue. Nice work. Best luck.
How to maintain the hypoxia condition?
We have maintained hypoxia condition by covering whole tank. In case of normoxia, we have given the continuous aeration, but in case of hypoxia, we entirely covered.
Okay.
Hello. Hello, sir.
Yes, ma'am.
May I present, sir?
Yes, ma'am.
Oh, just 2 minutes.
Okay.
So my PPT is visible.
No, ma'am.
Just 2 minutes. Support. Yeah.
Please share your PPT, ma'am, firstly. Your screen is visible, ma'am. Please unmute yourself and start your presentation. Please unmute yourself, ma'am. Hello, ma'am. Hello. Please unmute yourself. Hello. Jyoti Pulpagare, ma'am. Hello. Hello. Jyoti Pulpagare, ma'am.
Hello. Hello.
Yes, ma'am.
I'm audible, sir.
Yes, ma'am. Please start your presentation.
Okay, sir. Respected chairman and co-person, and my research topic is about the topic to find out of some station from Sakri Tahsil, Disdule. My abstract is general survey was carried out to appreciate the diversity of fishes from 4 different stations like Sakri, Kasare, Dehvel and Pimpalner during June 2020 to 2024. Throughout the study, total 19 species were collected, which belonging to 6 orders, followed by 10 families and 18 genera. Orders Cyprinforms were dominated with 11 species and family Cypernidae with 10 species. Authors documented the record of fish diversity by calculating the various diversity indices such as Shannon Weaver diversity, simple dominance index, Shannon index of diversity, Simpson reciprocal index, Margalful index, and Petulos-Gibbons index. Introduction. In 20th century, from last few decades, fish is a natural renewable resources as only in terms of total variety, but high quality animal protein food. Globally first provide after 16% of the animal protein documented by human because high biological value. It contains all necessary amino acid in pleasuring quality. Flesh of fish include vital components in various quality like carbohydrates, protein, fats, ash and water. In healthy fishes, averagely contains 72% water, 19% protein, 8% fat, 0.5% calcium, 0.25 phosphorus, and 0.1% vitamin A, B, C, and D. Low fat content and low caloric value. This is a review of literature like Patole and Jadhav, Sonone and Patole and Borane. We use a material and method like physiographic area to choose for this research like Sakri, Kasare, Dahivel, and Pimpernel. The healthy, proper size fresh fish specimen were purchased from fishermen at the site of study period. The collected specimen were immediately covered with the ice chest for keeping freshness. For identification and taxonomic study, the photographs were snapped. The fishes were identified by using different literature like Day, Talwar, Jingargran, and Jairam. Data collection. For estimation of diversity indices, total number of species, total number of individual, sample, and number of individual of species were recorded. 6 types of diversity indices survey were used, like Shannon-Weaver Index, Simpson-Dominant Index, Simpson Index of Diversity, Simpson Reciprocal Index, Mangelsly Index, and Pilot's Evenness Index. Result and discussion. General survey was carried out to evaluate the diversity of fishes from 4 different stations like Sakri, Kasari, Devel, and Pimpernel during June 2020 to May 2024. Total 19 species were collected, which belonging to 6 orders, followed by 10 families, 18 genera, and species. Orders Cypriniformes were dominated with 11 species and family Cypernidae with 10 species. The specimen have washed with tap water to remove the debris from clearing the mucus. Small specimen were directly placed in 4% formalin, where large species were given an incision in their abdomen and preserved. The fishes were identified by using different literature, Day and Talwar and Jairam. Those which are not properly identified were sent to Western Regional Station. This is a fish observed from different stations during June 2020 to May. There is a scientific name of fishes and common name. There is a Sakri, Kasare, Dahivel, and Pimpernel station. Collected fish specimen. Family-wise percentage of fishes from Sakri. 60% is a Cypernidae, 20% Bagiridae, and 20% Nemachilidae. Family-wise percent of fishes from Kasare, 50% Cypernidae, 17% Chichidae, 70% Canidae, and 60% Master Semi-Gridae. Family-wise percentage from Devel, there is about 34% Bagiridae family, 33% Notoperidae family, and Cypernidae family belongs to 33%. And last is from Pimpernel, we were collected 34% Celluloidae fish specimen, 33% Cypernidae, and Clupidae 33%. Fish specimen richness, abundance, dominance, and diversity indices. Like species richness is about 90, species abundance 242, Simpson dominance index is 0.0712, Simpson's reciprocal index is 13.0, and Pilot's evenness indexes is 0.5251. The present investigation focus richness, abundance, and different diversity indices for 4 different freshwater bodies. The lake represent richer value about fish species, that is 19 and abundance 242 in 4 years study period. The value of the Shannon-Weaver index was, it shows greater diversity. The value of the Simpson dominance index where which ranges between 0 and 1. To get over this problem, D is subtracted from 1 to get the Simpson indexes, which was Simpson reciprocal index, that is 1 by D. This there was the closely related indices, but the value of Simpson dominance index and Simpson index of diversity shows increase in status. Our findings are supported by above studies like Sonone and Patole, Jewell and Cheng. Conclusion about this research topic is based on the result of this research. It was observed that diversity of fish fauna is more in Kasari village as compared to remaining 3 stations. It is recommended that further the reservoir can be considered being good condition. Hence, there is an urgent need to create awareness among local people on importance of reservoir habitat and its fish fauna and the need to conserve them for future generation. There is a references related to research topic. Thank you so much, sir. This is my presentation.
Okay, madam. What is the reciprocal index?
Reciprocal index is how much fish specimens are comparatively found into 4 different areas, and there is a high percentage or low percentage, and then we
Okay.
conclude the reciprocal index.
Okay, okay.
Hello, sir. Hello, sir. May I mute?
Yes, ma'am.
Yes. Thank you, sir, very much.
May I go next, sir?
Yes, ma'am. Can you present?
Yes.
Okay. Start, ma'am.
Is the screen visible? Hello?
Yes, yes.
Yes, ma'am. Please start your presentation.
Yes. So, good morning, everyone. I am Ojas Birari, and I am a second year studying zoology at Dr. D.Y. Patil Arts, Commerce and Science Women's College in Pimpri, Pune. Today, my topic of presentation would be the potential of mint plant extract as an eco-friendly larvicidal agent for houseflies. So, as everyone is aware, houseflies are one of the major domestic and veterinary pests and vectors of various diseases worldwide. It is because of their ability to exploit most ecological niches due to their larvae's ability to evolve and hence cause quite a lot of problems. Now, to curb their growth, we usually do use chemical insecticides. But these, as previously mentioned in the keynote speech, are these insecticides cause various health concerns and pose a threat to environment. So, as a better alternative, eco-friendly insecticides could be used as they are degradable, species specific, and have zero side effects on non-target organisms. So, for the research material methodology, we cultured housefly larva by collecting the adult flies in. Hello, am I audible?
Yes, ma'am. You are audible.
Okay, so the eggs were incubated in plastic jars and on moist groundnut just crushed powder in the laboratory at 28 degrees Celsius and were allowed to develop larva and into pupae on the same medium. And then the pupae were transferred in other containers to for adult emergence, which were then cultured. These were how we were collected in the containers, and the lid was covered with muslin cloth in order for them to breathe. And according to the life cycle, it it usually takes up to a day for the larva to emerge from the eggs and 30 days for the larva to turn into pupae and then adults. So for the plant leaf extract, we took fresh mint leaves and then we shed dried them for 6 to 8 days at 30 to 32 degrees Celsius. And the dried leaf powder that was extracted, it was then extracted in menthol solvent in a sock-slit apparatus, and then they were dried using a rotary vacuum evaporator. So from the observations, first we separated the first and third instar larva in 5 groups of each in 5 groups of 15 larvae each, and repeated the experiment 6 times with the extract we prepared at 1, 2, 3, and 1, 2, 3, 4, and 5% of the extract. And according to observations, the first instar larva were more susceptible than the third instar larva. And according to the results, the lethal concentration value was at 1.5% for the first instar larva, whereas the lethal concentration of of value 50 was at 2% for third instar larva. So the different concentrations of extract were used to obtain mortality against first and third instar larva of housefly. In control, the first and third instar larva remained alive. The first instar larva showed higher mortality percentage compared to third instar larva as the concentration of extract was increased from 1% to 5% gradually. Based on the results achieved, the Mentera peperita extract shows larvicidal activity towards Muca domestica larva. These
that they can generate a auth token, and that auth token gets passed on to my basically.
Could you please repeat, sir?
So there was a ticket PM 1514. In that there is subtab which basically is talking about this auth token and everything. This was the not token.
Your voice was faint. Could you please repeat?
So go and take from it. I had a discussion with Hiren yesterday, but that is something that we need to check on. I also tried to check with Sandeep, but I think he also is not completely aware. Maybe it was long time back, so people tend to forget. So we have to look back at the code and figure out what was done exactly percentage.
I think there is a problem with my mic. I cannot hear clearly.
Sir, your voice is breaking, not clearly here.
Okay. So you will do this in yeah.
Should I continue till then?
Yes, madam. Yes, continue.
Okay, so as you can see in the picture, the larvae on the left side are were the ones that were kept in control, and on the left side, on the right side, are the larvae that were infected or affected due to the extract. Similarly, these were the results that are observed. And through this, we come to the conclusion that these extracts of mint leaves have promising effect as larvicidal control agents. These plants, being biological, are not toxic to mammals, and therefore they are quite harmless and could be used as potential natural insecticides. The most striking fact is the simplicity of the method of application, which is suitable and economical for controlling housefly in most breeding sites. And it can be concluded that the leaves can be useful in control and management of the housefly population and transmission of various pathogens associated with it. Thank you.
Okay, only one question is there.
Yes.
What is the active compound of that mentha?
It is menthol.
Menthol.
Yes. So
Okay.
it actually attacks to the senses of the housefly, which then causes a breakdown in its structure, and they shrivel up.
Okay. Okay, continue.
Yes. Any other questions?
I think there is no question, ma'am.
Yes, thank you.
Welcome, ma'am. Anyone ready for presentation?
Hello, sir.
Go
May I
ahead.
present? Hello, sir. This is Shubhada Fadnis. May I present my screen?
Yes, ma'am. Yes, ma'am.
Thank you, sir.
Welcome. Please start your presentation. Share your screen.
My screen is visible, no, sir?
Yes, ma'am.
Yeah, thank you, sir.
Please start your presentation.
So, good morning, everyone. My name is Shubhada Fadnis, and I'm presenting abstract on topic that is effect of few botanical extracts on mango pest, mango leaf hopper, and thrips. So, this is my abstract. Basically, Alfonso is the most popular and delicious leading cultivar. Alfonso is the most popular and delicious leading cultivar of mango, which is commercially grown in South Konkan region. Although the mango cultivation in Konkan region is vast, the productivity is very low as compared to the national productivity. So mango is vulnerable to variety of pests, including insects, mites, various pathogens, viruses, etc. But the insect pests are a major limiting factor in achieving full yield potential of the mango varieties. Among the mango insect pests, mango leafhopper and thrips are major serious and destructive pests of mango. Those scales, mealybugs, mites, leaf weber, stem borer, white flies, etc., were earlier considered to be minor or the secondary pests. Now they have become serious problem in recent years. So to mitigate this problem, the control of the insect pests in agricultural scale means still it is very challenging. So widespread use of the synthetic organic insecticides in orchards pest control has resulted in damage to the environment, pest resistance to the insecticides. By considering all these facts, present study aims to evaluate biocidal activities of locally available plants. And here are the yeah, here it is the aim and the objective of my topic. So to study and identify the local Hello, am I audible, sir?
Yes, ma'am. Yes, ma'am.
Yes, ma'am.
So aim and the objectives of my topic is to study and identify the local weed flora, which is easily available and non-toxic to non-target organisms. Is to study effects of phytoextracts using locally available plants to control major mango pests like mango leaf hopper, mango mealybug, thrips, whiteflies, aphids, etc. To investigate phytoextracts activities of the locally available plant species such as Ageratum conizoides and Chromolina odorata. So here are the material and methods. Basically, preliminary survey which was conducted, a seasonal survey and the taxonomic status of the mango insect pest. The collection of live mango insect pests from the known mango orchard and their rearing under controlled laboratory condition was carried out. So here you can see in the field work, at the time of field work, I collected mango leaf hopper from the mango orchard in district Ratnagiri. The collected mango leaf hoppers from the plants or the mango plant from the certain areas like a slope and the roadside areas where the mango leaf hoppers on the basis of their size they're having various varieties. Their Hello, am I audible? Hello.
Yes,
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 ma'am.
ma'am.
Okay. So mango leaf hoppers predominantly puncture and sucks the entire sap of that plant or the inflorescence or tender shoots. And so that I collected these hoppers in the seasonal survey. At the same time, in the field, mango orchard, the high incidences of thrips were also observed. The thrip specimens were collected from the mango orchard located in Gavhani district, Ratnagiri. Both the young and the adult insects, thrips, you can see here in the picture on the mobile screen. These are very sensitive to light, so collected against the light on the mobile screen. Um, During this process, the rearing of insects using various hosts is very important to understand their biology and behavioral pattern. So mass rearing for a couple of generation is critical when the insects are economically very important, just like the mango leaf hopper. So I tried one trial that an attempt was carried out to rear the mango leaf hoppers in Pune. The climatic conditions play an important role in the appearance of mango leaf hopper, that is highly adaptive to the humid and the hot conditions, as well as they breed and feed on tender shoots, inflorescence, panicles, and very tender leaves also. So that this trial found to be unsuccessful here in Pune. Um, as you see on the screen, the basic survey was carried out across the district Ratnagiri on locally available weeds which have insecticidal properties. The following plants have been observed abundantly, so proposed to select for the study of the pesticide property. First one is the Ageratum conizoides. and this second variety, that is Cromolina odorata, which is abundantly seen in and around the field area. The Ageratum and the Eupatorium species were selected for the entire checking the pesticidal properties. These are the following reasons for selecting these plants. The plants have been observed abundantly. Second thing, the herbal pesticide properties are seen in these plants, and these plants are very easy to collect. So the plants are collected, the leaves are collected, air shed dried, as you see on the screen, and air-shed dried plant material in the powder form is kept ready using mixer grinder. So after the standardization and the remaining processes, here I carried out the experiment with the help of these powders with the decoction. The lab experiments were carried out by the leaf dip method, film smear method, with the 3 replications of each, and the observations were taken time to time. So here are the results. Ageratum conizoides is effective against mango leaf hopper in bioassays. The leaf dip method exhibited more positive effects than the film smear method. All these readings in the table, these are the values, are average of 3 determinants. So here is the result and discussion. The results show that the leaf dip method exhibited more positive effects than the film smear method. Ageratum conizoides effective against the mango leaf hopper in bioassays. The current study implies that in place of synthetic insecticides, biopesticides are an eco-safe alternative. Biologically active natural plant products may play a significant role in this regard. So these are the references I reviewed during my studies. Yes, thank you. And this is all about my topic.
Okay, only one question is there.
Yes, sir.
Uh, which solvent you used to treat the paste?
Sorry, sir, I can't hear you.
Solvent used by you?
Yes, I use the solvent, that is aqueous solution, to prepare all the concentrations.
Acetone?
No, aqueous solution in water,
Okay,
water-based solution.
okay. And what is the active compound?
Active compounds are under processor, but the tannins, alkaloids, flavonoids, these are the compounds found in the plant material, in leaves and the seeds, in my review papers, while
Okay.
reviewing it.
Okay.
Yes, sir. Thank you.
Pooja, may I go next?
Anyone can ready for presentation?
Hello, my name is Rutuja. May I go next for the presentation?
Yes, ma'am. Yes, Rutuja, ma'am. Please share your screen.
Is my screen visible?
No, ma'am. Not yet visible.
Now, is it?
No, ma'am.
Just a minute. Sir, is it visible now?
No, ma'am. Please share again.
Okay, just a minute.
I think ma'am is left. Anyone can next. Anyone can ready for presentation? Hello. Anyone can ready for presentation?
Hello, sir.
Hello.
Hello, sir. Am I audible?
Yes, Ashwini ma'am.
Sir, can I present now?
Yes, ma'am. Please share your screen.
Okay, sir. Just give me 2 minutes.
Yes, ma'am. Yes,
Is my
your
screen visible?
yes, ma'am. Please make a screen share. Yes, yes, ma'am.
So, good afternoon, one and all. Myself, Ashwini Deshkar. So, my topic means I'm participated for poster competition. So, related to my research work, after reviewing some of the literature, what topic I have decided that one is a review on verticillium species to decide bless or a curse. The increase in the world's population from past few decades demand equal increase in food production. Maximum agricultural crops produces yield depending on the application of pesticides. Pesticides are the substances used against pests which are harmful to the crops. So nowadays pest problem is a major obstacle in obtaining higher yields out of the targeted crops in India as well as all over the world. Being agricultural country, India loses about 30% of its crop yield due to infestation of pests and various diseases. This damage due to attack by pests estimated to be 50,000 crore and above annually. Pesticides used to kill the pest as well as the insects which attacks on the crops. Different types of pesticides have been used for the protection of crops. Use of pesticide have a positive impact over the pest by controlling them as well as it has a negative impact on non-targeted beneficial insects. Beneficial means the phytophagous one, the predatory one. So there is a need to use, so even government is also insisting to use the biocide instead of pesticide. So my research work is related to verticillium species, and while reviewing some of the things which I have noticed, so in the methodology, the verticillium licheni, this is a naturally occurring fungus that can be used as a biological insecticide to control insect pests in agriculture as well as horticulture. It is effective against aphids, mealybugs, and scaly insects. The infection which you will see, this I have spotted during my collection. Verticillium, the another species of Verticillium delhi and Verticillium albo-ultra causes the disease verticillium wilt. Verticillium delhi spores, these are commonly called as verticillium wilt. It's a disease, fungal infection, which can be produced with the help of spores, microsclerotica. The spores of verticillium are small, hard structures that can be withstand a variety of conditions, including high and low temperature and dehydration. These spores are survived in soil for many years without the host. So, after comparing these two, means at one side verticillium licheni like biocides, they are effective against various pests. They can act as the predatory one, and after revising this verticillium delhi and verticillium albo-ultra, they causes the disease verticillium wilt. So there is need to do the research related to verticillium species and to conclude whether that can be used as or that can be called as bless or curse. Related to my work, few references I have studied. Out of that, these are some of them. Thank you. Am I audible?
Yes, ma'am.
Thank you, sir.
Okay, ma'am. Next, Ankita ma'am, are you here?
Yes, sir.
Can you present your research?
Sure, sir.
Okay, then share your PPT.
Sir, I am visible.
No, ma'am. Yes, ma'am. Yes. Please
Good
make.
afternoon, everyone.
Hello, ma'am.
Huh? Yes, sir.
Please make. Yes, yes, yes. Okay, okay. Yes. Full screen. Yes. Okay, start, ma'am. Please.
Good afternoon, everyone. My name is Ankita Prasad, and my today's topic for presentation is role of microplastics in altering ecosystem services and evaluating its ecotoxical effects. And this is a review which I am going to present today. And I am working at Department of Zoology as a PhD student under Shinde sir in KJ Somaya College, Kopargaon, which is affiliated to Savitribai Phule, Pune University. My background of the study is the concerning problem of increased amount of plastics which have been manufactured in high quantity. In 1950, the amount of plastic manufacture was about 2.5 million, which is now increased to 448 million tons just in 2015. And the microplastics are the plastics which are plastics of very small size, less than 1 micrometer to 5,000 micrometer or 5 micrometer. There are mainly two types of microplastics: primary microplastics and secondary microplastics. Primary microplastics are the tiny plastic bits which are intentionally, deliberately manufactured in that small dimension, and secondary microplastics are formed by the degradation of bigger microplastics materials due to the environmental factors. The primary microplastics are manufactured so as to incorporate them in the daily life materials like cosmetics and face washes, shampoos, toothpaste, etc. And secondary microplastics are the plastics which are broken down into smaller pieces due to the environmental effects like wave action, wind action, and temperature, UV radiation, aberration, etc. And ultimately, they get spread into our atmosphere, terrestrial ecosystem, and most importantly, in our aquatic ecosystems. The plastics the plastics are increasing in our environment due to their high manufacturing and low recycle rate. And the plastics of microplastics are found in various sizes, colors, and chemical composition. And they are found in the fibers, fragments, films, biofilms, foams, sheets, flakes, pellets, etc. And most abundantly and frequently observed microplastics are the fibers, which are seen in 80% of the studies. and microplastics in which fibers are most abundantly found, and the most abundant areas where these fibers are found in the organisms are the gut and the liver of the body, which is reported till now in 57% of the reports. These microplastics block the digestive tract, increase mortality, decrease the fecundity, lead to the inflammatory responses, alter our metabolism, disrupt reproduction, change in behavior, decrease energy reserves, causes oxidative stress, decrease mobility in some species. There are also reports that shows the gene expression disruption and damage of reproductive organs, growth inhibition, retardation, reduction of weight, disturbance in antioxidative conditions of liver, etc. In addition to microplastics, there are various toxic monomers or additives which are added to the microplastic, which also have the toxic effect, and the plastics may also act as a vector for various other contaminants like heavy metals and other pathogens. These are the various different famous types of plastics which are commonly used, and these plastics get incorporated into the water body due to the domestic waste, agricultural waste, industrial waste, and flooded waters. Microplastic have ability to impact the water quality and in turn influence the behavior and physiology of different aquatic animals, and the exposure to microplastic may lead to the reproductive responses and decrease in the quality of gametes and oocytes, sperm swimming speed, fecundity, and offspring quality. The consumption of microplastic, while large filter feeding megafauna such as baleen whales, the filter feeder sharks, and sea turtles present a possible threat to endangered species. The microplastic accumulate and magnify, means they bioaccumulate and biomagnify in concentration along the food chain, and this magnification and accumulation uh, uh, leads to have different factors due to which they accumulate due to the body weight, habitat, and food sources of the organism which are consuming them. The trophic transfer is the primary means by which a big filter feeder is exposed to the microplastic contamination rather than a tough direct ambient contact. The whales are believed to consume approximately 3 million microplastics on the daily basis, and baleen whales predominantly consume food at a depth where microplastic concentrations are high, significantly between 50 to 250 meters. The interaction between baleen and plastic causes multiple risks to the whale, such as occlusion and damage to their filtering systems, which can hinder their ability to feed. And there is greater threat to certain species of whales, such as the right whale baleen and humpback baleen, due to various factors, including porosity of their filters, their diet, habitat, and geographical distribution where they are foraging, and their ecology and behavior. The accumulation of microplastic along the beaches and shores affect the breeding habitats of marine turtles, seabirds, and other species that inhabits in the coastal areas. And this consumption of micronizing plastics by newly hatched sea turtle poses a significant threat to their survival. A research conducted on unviable loggerhead sea turtle eggs discovered the presence of microplastic in underdeveloped sea turtle eggs, which indicates the potential transfer of these small particles from mother to her offspring. And these consuming microplastic may also lead to the blockage and injuries to the intestine, dilution of diet, starvation, malnutrition, and increased buoyancy. These effects result in negative health outcomes such as reduced growth rates, reproductive output, and overall health, and can even lead to the mortality in the sea
स्लाइडचे
turtle.
फॉर्मॅट बघायला भेटते.
These affect the both temperature and permeability of sediment, which is nest of the turtles, and affect the hatching and reproductive success, sex ratios, and productivity of the nest, sexual development, and other factors, due to which there is a danger to the marine turtle populations. And disruption of natural environment resulted in substantial influence on the foraging and nesting behavior of birds, and they consume the plastic fragments, mistakenly seeing them as their food. And it is observed that there is prevalence of medium size and small size meso and microplastics in their gut, and these leads to the stomach blockage and punctures. Birds have been observed to incorporate these plastic materials into their nest for structural, defensive, and insulating purposes, and which is linked to entanglement and potential fatality of nestling due to the stranglings. The adverse impact of micro and microplastic on the birds leads on the physiology, growth, development, breeding success, and survival of the birds have also been recorded. In reference to the terrestrial environment, the microplastic leads to the pollution and also pose the possible threat to soil health, organisms, and ecosystem, have detrimental impact on the soil biodiversity and microbial communities of the soil, nutrient cycling, and overall ecosystem functioning. They have potential to influence agricultural output and altering the soil characteristics and nutrient availability, which in turn affects the plant growth and development. These microplastic built up in the sediment and disrupt the nutrient cycling, and also affects and have the deterioration effect on the habitats like coral reefs and mangroves. And these may accelerate the ecological these may accelerate the negative impact on the ecological services like carbon sequestration, food and water supply, coastal defense, marine habitat, environmental purification, and climate regulation. There are various benthic organisms like crustaceans, mussels, and bivalves which live on the coastal region, and the 90% of microplastic which are trapped in the sediment may lead to damage to the tissues. And the coral reef which consume or accumulate the microplastics in them have the significant damage to their tissues, compromises their immune system, reduces their food consumption, hinders their growth, and causes necrosis and bleaching, and also leads to the decreased calcification and ultimately affecting the growth of coral skeleton. And these microplastic have ability to be carried across vast distances via air and water currents, which have an resulting extensive environmental damage effect. And these microplastic are significant threat to marine, coastal, terrestrial, and avian biodiversity and ecosystems. And and overall microplastic can disturb the equilibrium of ecosystem by impacting the distribution and population size of specific species, as the certain species are more vulnerable and susceptible to the levels of microplastic pollution, which in turn impacts the ability to survive and thrive. And these studies are have been done on various different organisms till now, from zooplanktons to poriferans, crustaceans, mollusks, fishes, amphibians, and various different group of organisms, and various different studies have been done till now. And but these studies are very rare and very less in India, and the most of the studies are done in the marine water and the freshwater, terrestrial, and human consumable things, and atmospheric studies are very rare in compared to the marine ecosystem studies. And if we see about the data, the most of the data arises from the northern and eastern part of the India and southern part of India, and at the central part of India there are very rare or no data available for these microplastics. And only one paper is available from NIT at the Andhra Pradesh from our Godavari river, which have sampled the water of Godavari river at 5 different sites, and mainly they focused on the shape of the microplastics and color of the microplastics which are found in the Godavari river. And this is also the at the end point of the Godavari river, at the place where this study have been done, and at the origin of Godavari in our Maharashtra area, there is very rare, or I have not found any studies on the Godavari river on the microplastics. So I am currently studying on this topic, and thank you. Any questions?
Only one question is there. What is baleen? Is it
Baleen,
audible?
baleen
BAN?
is the organ of the whale, and baleen is the main. Just a second. Baleen whale. I can show that. This portion of whale is the baleen where it feeds, filters the feeding.
Okay.
This portion.
Thank you. Proceed for.
Can anyone ready or present your research?
May I go next?
Yes, ma'am.
Is my screen visible for the presentation?
Yes, ma'am.
Okay, thank you. My name is Rutuja Ashok Nigod. I'm a faculty at SIS College, Sion West, Mumbai, in the Department of Zoology. First of all, good afternoon to respected judges, distinguished guests, and fellow participant. So I'm going to present my research on the topic effect of hypoxia on the protein content in fish. So these are going to be my content where I'm going to discuss about the objective, materials and methods, followed by results and discussion, conclusion, and the future prospects. So as you all know, kind of in the recent decades, anthropogenic interventions such as your agro-fertilizer waste, domestic sewage discharges, etc., have exponentially increased nutrient inputs from the original level, leading to eutrophication. Also, if we look into this matter, dissolved oxygen, which is an important and essential component, which kind of determines the water quality and also the tropodynamics of an water aquatic system. It was declared by the US Environmental Protection Act that in any healthy aquatic ecosystem, the concentration of oxygen must always be for the proper growth and survival of the aquatic ecosystem. It should be somewhere around 4.8 milligram per liter. But when this criterion is kind of
we usually see that the fall of oxygen below this chronic criterion, it is referred to as hypoxia, which we also see that has an adverse effect on the health of the aquatic system. Hypoxia in aquatic systems so basically refers to waters where the dissolved oxygen concentration is below 2 mg per liter of water. So basically in this study, we kind of made an attempt to study the effect of hypoxia on protein content in fish, where we used the animal model, which was a catfish. The scientific name of this catfish is Heteropenesis fossilis. Now, the reason behind choosing this animal model was that, particularly in the Asian region, it is economically important edible fish, and its flesh is also very rich in protein and iron. So we wanted to we wanted to carry out a study wherein we wanted to see the effect of hypoxia on the protein expression on the various organs. So in this present study, we basically focused to see the changes in the fish protein content in different tissues such as brain, ovary, muscles, liver, gills, and kidneys, which were kind of uh, exposed to uh, acute hypoxic period, which help us to understand the changes in fish metabolism and also the organ function in response to the environmental stresses as the mechanism of adaptation. So this was our overall objective. Coming to the materials and methods, this was the experimental design where we had two tanks designed. One was for normoxia, which was quietly good aerated, and the other was for hypoxia, where which we had covered with proper lid, so there was no contact with the atmospheric oxygen. The only dissolved oxygen was the source for the fishes. They were there in the hypoxic tank. So the primary behavioral changes or the morphological changes that we observed during the experiment, as you can see in the diagram, they were the formation of bubble in the tank. Also, we could see intermittent ventilation as an escape to hypoxia. Also, the fishes showed decreased activity where they were completely lying down, and there were not such movement were seen in the fishes. Now, coming to the protein estimation, we basically did first protein extraction, which was followed by protein estimation. So for the extraction of proteins, this were the tissue samples we had dissected from the catfish, and we had homogenized in a PBS buffer under cold conditions, and then later it were centrifuged for around 25 minutes, and then we had taken the supernatant for further analysis. As you can see here, as I told, that the protein extraction was followed by protein estimation. So we here used Lowry method. So we had to basically test tube stand. One was for blank, and for the tissue samples, where the tissue samples from the normoxia and the hypoxia groups were estimated according to the Lowry method. The supernatant that we had collected, we mixed it with a reagent C, and then we incubated for somewhere around 10 minutes, and then we added Folin Katau reagent, and then we analyzed it, measured it at 750 nanometer using a digital spectrophotometer. So coming to the main part of this research that was conducted was we could see variation in the protein expression of the different tissue organs. The significantly protein expression were reduced when it came to liver, gills, kidney, muscle, but it was only in the brain where we could see that the protein expression was elevated when it came to hypoxia. Rest of the organs, we could find out that the in hypoxic groups, the protein concentration was significantly reduced. So significantly reduced, and therefore as you can see, this reduced protein concentration in different tissue organs had various adverse effects on the fish with respect to osmoregulatory impairments, physiological impairments, where the feeding mechanism was altered, the growth, the survival, all these parameters were altered. Metabolism impairments were also seen. Also, you can see the fish were exposed to hypoxia. Therefore, there was a stress response or stress-induced proteolysis was seen, and this proteins they were catabolized, and also further analysis suggests that gluconeogenesis was also considered as a pathway to kind of use energy source to avoid hypoxia. Also, due to the reduced protein expression when it came to ovary, there was significantly correlated with reduced reproductive functioning. Although when it came to brain, there was an increased protein content in brain, because when it comes to brain, it is a very hypoxic sensitive organ, and therefore to coordinate with the down regulator or with the signaling pathways downways, there were the neuroglobulins, particularly in the brain, they should increase protein concentration. And therefore, when it comes to the future prospect, as we know, this anthropogenic activities, this has a which are kind of increasing the hypoxic environment in the aquatic ecosystem, the pollution, all this has an effect on the aquaculture, on the productivity of the fish, wherein the hypoxic, the fishes therefore have evolved various physiological, anatomical adaptation to kind of escape from the or to avoid hypoxia. So they are using various several mechanism as an adaptation towards hypoxia. And these are my references. And lastly, I will thank my head of the department, Dr. Sati Sarfare, who provided us the facilities, and my research supervisor, Dr. Surbi Mishra, for helping me out throughout this research presentation. Thank you. My session is open for discussion.
Okay, what about soluble protein?
Sorry.
What about soluble proteins?
Soluble proteins in respect to
Your work is only concerned.
कट करो.
Hello.
We generally focused on the protein expression in the tissues. So soluble proteins, as we know, can be involved in regulating hypoxia, and therefore there may be some and due to hypoxia it can happen that proteins can become kind of insoluble also. So that this can be one of the impact of hypoxia, I feel.
Okay, continue.
Should
Hello, I
I
have one
stop?
question.
Okay.
You use the terms evolution and adaptation simultaneously. So is it adaptation or
No,
evolution?
I just use the word adaptation. I didn't use the word evolution. Hypoxia is therefore used as an adaptation by the fish, I said.
For your last slide, you use the term evolve. The fishes evolved like that. So I want to just clarify. It is not a question, but I want to just clarify: is it evolved or adapt?
It is adaptation.
Okay, thank you then.
In case of male, similar pattern is available or not?
Sorry.
You work on the female only. So
Yeah.
do you any idea about the male in the same?
So the cats that we got, we were so most of them were
But there is any
female.
criteria for choosing the particular female? You may select also male also. You particularly select female. So there is any criteria on which base you select only the
No,
female?
there was not such any criteria. It was actually the availability of the fishes, and whatever the fishes we got, they were mostly female. So we decided that we'll choose gonad in the form of ovary as one of the organs.
Okay.
So I feel if there is an impact on this on ovary also, which has an impact later on the reproductive functioning. Obvious, there may be also effect on testes also, which
That's
will
testes also is a reproductive organ. So the any
we
kind of change in
we
testes,
have
they
not
also
actually
ultimately
carried
the
study.
Okay.
We have not actually carried study on the testes, but on should I stop sharing or any other questions?
याला कसं आहे
Can anyone ready or present your research work? Hello. Hello. Can anyone ready or present your research work?
Anyone is ready with your presentation? Hello. Zamdade Ganesh, are you ready with your presentation? Dr. Zamdade.
Hello. Now this session is over. We again resume at 1:30 for plenary lecture.
Okay, thank you.
Thank you, sir.
Okay,
Thank you,
thank you.
one and all.